Welcome everyone to the third webinar this year from Confocal NL. Today we will be talking about organoid research to study organ development and model human diseases. We will start with a talk from Dr. Daniel Krueger. He's a postdoc at Utrecht University in the Netherlands, part of the famous Hans Clevers group. After this, we will have time to answer some of the questions you've put in the chat. Hereafter, we will have a short demonstration of the NL5 by Anna Veher, who is the product application specialist at Confocal NL. And then ending with another question and answers round. We expect this webinar to take approximately 45 minutes. So without further ado, I would like to give the word to Dr. Daniel Krueger.
Thanks. Okay. All right. Yes. Welcome, everyone. Thank you for joining. Yes, I will just give a brief overview about what we do actually in Hans Clevers' lab in terms of research regarding or using organoids as a vehicle, as a model. On the one hand, for studying biology, basic biology question, but on the other hand, also to model human diseases and as actually a vehicle to study, for example, drugs and drugs efficacy. And yeah, but to begin with, organoids are really like miniature versions of the big organs that we have in our body. And organoids can be produced in vitro in 3D structures and show kind of a very similar and realistic microanatomy compared to the real host tissue. And they're derived from stem cells that can be, on the one hand, embryonic stem cells, but originally identified was actually that that they can also be derived from specific organs, and here basically from the adult stem cells that actually sit in each of these organs and give rise to kind of a big diversity of terminally differentiated cells, for example, cells of the gut, of the small intestine, of the pancreas, etc. And of course, I will not focus on any of that, but terminally differentiated cells can be also reprogrammed to become more embryonic stem cell-like. This was identified by Krueger Yamanaka, and also then like terminally differentiated cells that were brought back to this embryonic stem cell state can also produce organs and mimic kind of the environment and in vivo situation. But at the basis of organoid research was really a key finding, and that was actually the identification of the first adult stem cells in the gut, in the human gut. And the critical step was really the identification of a marker for intestinal stem cells, which is called LGR5. So LGR5 is a gene that is very specifically marking, as you can see here, hopefully you see my mouse, it very specifically marks the bottom of the intestine. So here in this picture, what you see here is actually a region that is called crypt region, and these very long villi that line basically the intestinal epithelium, so the inside of the gut. And it was already known before that this bottom part is really the origin of cells, but the identification of a marker that specifically labels these adult stem cell compartments was a very big step, and was actually found by using a very elegant set of experiments that these cells that are labeled here in green can actually produce all the other cell types that you find in the intestine till the very tip of the villus. And the next key experiment for the generation of organoid, as we know it today, was then that you actually can extract these cells based on this cellular marker, and grow actually these single cells in a specific environment that kind of mimics the situation in vivo So where we have basically tried to to recapitulate what is happening in terms of growth factor, environment, and so on in the intestine, in the human body, or in the mouse, and you provide these factors, and this actually allows these individual adult stem cells to grow and form structures that remarkably actually represent different aspects of the adult organ. And this was really the initial stage. So the first organ that was grown, these so-called mini guts. But of course, by now, actually, organoids from all different kinds of tissues were actually grown and were successfully generated. For example, from liver, pancreas, lung, stomach, and so on. And they all kind of have similar key features, which is on the one hand that you can really grow them from single adult stem cells, but also what is very important is that they are very genetically stable, and that they actually give rise to a wide set of terminally differentiated cells. So it's not only that you have one cell type as in classical cell culture, but instead they actually produce a lot of different very specialized cell types. And you can almost grow them indefinitely, so you can expand them a lot and do a lot of experiments. And they all, always grow in these kind of matrix in a special gel, which really allows for 3D growth, that you can really analyze these things as if you would kind of have a spotlight in the human or the mouse body. And that, of course, opens a wide avenue for all kind of experiments that were basically before the development of organoids not possible. So they provide new prospects, for example, for generative medicine, for drug development and therapy, but also for basic research and developmental biology. And I want to focus basically on three different aspects. One is how we model certain human diseases in our lab, but also look a little bit of how drug discovery looks like in using organoids as a tool, and also to look to see actually how can we actually look at developmental processes that were otherwise hidden in the inside of the body, pretty much, if you want to take it with these words. And yes, I think disease modeling is really a key aspect, a central application of organoid research. And our lab, for example, is part of the OnCode consortium, whose mission is really to understand better, on the one hand, cancer biology, and translate also this research into practice and eventually treat patients. And this is a big avenue, and it really aims to, for example, obtain a very comprehensive data set of organoids, for example, from cancer patients. But not only, and the aim is not only to obtain cancerous tissue growth in the lab, but also at the same time grow normal tissues from the same patient, so that you have actually a side-to-side, or that enables a side-to-side comparison of healthy and diseased tissue, which then, of course, allows much better, much more precise experiments when it comes to the development of drugs, or for example, if you think of personalized medicine, to really advise in the future therapy, for example, of a patient that has cancer, and you suggest, for example, a certain chemotherapy. Yes, and then kind of a very big research entity is actually, a big research resource is actually the hub that is based in Utrecht, was founded actually by people from Hans' lab, from Hans Klever's lab, and also from different other people, of course, but especially people from this Utrecht science community. And what they did was to actually open a resource, a huge biobank that contains hundreds of human organoid lines, as well as from normal tissue, from diseased tissue, and they provide actually worldwide a lot of these organoid materials, as well as procedures, for example, to how to isolate organoids and so forth. And this can be really harvested to study cancer biology, on the one hand as a research tool, but in the future, as I said, as a therapeutic use, of course, and a diagnostic tool. And how does such an assay look like? I just want to show you one example here. So this is, for example, a imaging-based assay that we are working on and establishing right now, where we have a very basic imaging in combination with machine learning algorithms that automatically track organoids seated on an imaging plate. And we follow those organoids over time, discriminate between living and dead organoids. And then basically this organoid line is a cancer line from head and neck patients. So it's kind of a tumor that generates in the throat, for example. And this line has been treated with a standard classical drug called cisplatin. And by analyzing these, the growth curves or their area obtained, the area of these organoids, you can actually get these growth curves, and you see, like, depending on different concentrations, how does this chemotherapeutic agent really affect organoid growth over time. And you can see then, can analyze basically those response curves over time, which is, I think, a very powerful tool to really look at the efficacy of certain drugs and really in the future give advices, for example, for treatment of patients. So that's the long-term goal. Another example how organoids are used to actually model human diseases is another project or another focus that Hans Klever's lab has, and that is based on a monogenetic disease. It's a disease that is inherited. It's actually the most common monogenetic disease in Western societies. It's called cystic fibrosis. This disease is characterized by the accumulation of mucus in the lung and in the upper airways. So basically, these patients actually cannot breathe very well because the flow of air is impaired, and also these patients suffer a lot from chronic and very acute, first acute and then actually chronic inflammations. And the reason for that disease at the molecular level is are mutations in a channel, in a chloride channel. It's called CFTR. That's the name of this ion channel. And basically, the function of this channel is that it opens in response to certain cues, also from the environment, exports chloride channel, which then starts basically a flow of water, so that actually the extracellular environment can be controlled by the tissue itself, and basically generates a healthy epithelial surface fluid covering the epithelium. And this is impaired in these patients, and about half of the patients that actually suffer from this disease have a deletion, so the same, let's say, genetic problem, the same genetic mutation, which can be treated with some medication relatively effectively. That helps them a lot, actually. But the other half of the patients actually have very rare mutations, and where no standard therapy can be applied. And this is, of course, a problem. Because actually it's for the patients is a big problem to test various diseases, because many of these diseases have side effects. These medications are sometimes very expensive, and you see actually if a drug is effective, you see it only after a very long time. And I want to present to you a case where actually it's the first let's say experiment, or this first set of experiments that really led to personalized treatment. And this boy, this young man, actually here in a Dutch TV show, actually was the first person that benefited from organoid research for his own kind of medical treatment. And this is based on an imaging-based assay that has been developed in Hans Klever's laboratory. And I want to explain the assay now. So the general idea is that these patients, cystic fibrosis patients, are functionally different from healthy epithelium, from the healthy tissue. And what has been found previously actually was that we can actually do a very simple assay that allows us to really monitor this the effect of this disease. And this is called and this assay looks the following. So when you generate organoids from healthy people that don't have mutations in this CFTR channel, when you give them a certain drug that is actually very similar to a that's called phoscodin, and it's very similar to actually cholera toxin. So the a toxin that is produced by bacteria that induces massive diarrhea. And healthy organoids, when you add this they activate their functional CFTR channels and induce, let's say, an influx of water. And as these organoid structures are closed, it's a closed epithelium, they start to swell because there's more water, more fluid basically pumped into the organoids. Now, if you generate organoids from patients that actually suffer this disease, cystic fibrosis, and you add this drug, nothing happens. So you see here the time goes up, but actually these organoids don't respond. And this technique actually to derive these organoids is actually minimally invasive. So it's a rectal biopsy. The patients hardly feel anything. It's actually very, very mild. And from this biopsy, actually from patients, this can be grown. And this was also done for this young man that I've introduced you to you a slide before. And now actually having this assay at hand and being able to score this so efficiently, we could actually test drugs on those organoids. And actually one drug was actually on the same patient was showing actually a very strong response. And basically the set of experiments, and this is only kind of the basis for the most critical experiment for this whole data set, was enough to really advise therapy for this young man. And he could actually take this drug and is now actually having a more or less healthy and normal life. So that is really one of the cases where I think the organoid technology already made a very big impact, certainly for the life of this one person. But as I said, drug modeling is certainly a very big topic for organoid research, but it also allows us to study other processes. We look at developmental biology questions that are difficult to address otherwise. And one study that I would like to present to you is based on the question of how cells derive from common progenitor and commit to a certain fate, and whether actually this fate switch is permanent or if there's actually any variation from that, if they can switch back basically. And these fate switching and commitment to a certain fate is of course especially important for tissues that constantly renew and regenerate, such as the intestine. And because when you think of it, like the adult stem cells, actually they give rise to a huge variety of different cells. On the one hand, there's this big line of absorptive progenitor cells and absorptive cell types. So they take off nutrients, and on the other hand, there are a lot of different secretory cells produced. Some of them have very specific and particular functions, and I don't want to go into any of these details, but we're studying actually pretty much any of those more rare cell types. But they also give rise to a huge group of cells that are called enteroendocrine cells, and these cells actually produce many of the hormones, actually most of the hormones that we have in the human body. So they are very, very important for many different things in many different processes in the body. And it is not clear actually how these cells that all produce a different kind of set of hormones, how they actually derive from this common progenitor line. So what all these hormone-producing cells have in common is really a progenitor cell line that is characterized by a transcription factor called neogenin 3. And to really look into how these cell types are produced from this common progenitor, people in the lab, in Hans Klever's lab, have done a very elegant set of experiment or a system, generated a very elegant system that allows actually to look at the time at which the cells form. And this system is called the Chronos system, and it's basically two different fluorescent proteins that are produced downstream of this transcription factor that is specific to these common progenitor cells, Neugenin 3, and the one fluorescent protein, neon green, forms very quickly. It folds very fast, forms very quickly, but is then destabilized by a PES domain. So it has actually a very short half-life time. And a second fluorescent protein, T-tomato, takes a lot of time to form, but then it is very, very stable, so it stays there forever. And basically what you see when you look at organoids derived from mice that express the system is, so this here you see basically one organoid, and here you see a green cell. And when I play the movie here, you see that it actually quite rapidly changes its color, which represents basically that the cell matures and gets a new gets into a terminally differentiated cell. And this color changing actually makes it possible to estimate or correlate the kind of the age of the cells and cluster them, sort them using a FACS, and after that actually it is possible to do single cell sequencing and learn a lot about these different terminally differentiated cell types with this extra information of time. And what we can learn actually from this experiment is, here this movie explains a little bit in brief what they found. So here basically you have first is a stem cell that gives rise to the common enteroendocrine progenitor, and first it expresses neon green, and then over time it also accumulates T-tomato, so it switches from green to yellow, and terminally differentiated cells are ready. And based on this information, you get time information. As I said, you sequence the cells, and you basically get information about all the different genes that are expressed in these cells at a given time point. And that allows you to really extract information based on the whole lineage tree, when these cells form, and at which point there's, let's say, a bifurcation for a special specialization, for differentiation, for actually undergoing a new or taking over a new fate. And I think this is a very powerful analysis, and shows actually the how effective actually also imaging can actually help in combination with organoid research and the accessibility of organoids to answer and to address those questions. So what I want to say here is really that it's that imaging and organoid technology in combination really allows us to really derive conclusions based on the observations that we can have. And another aspect that I want to bring in, because this is actually what I work on in the lab of Hans Klever's right now, is I want to test some of these conclusions that we have derived from observation, and actually test these conclusions by actually controlling the biology. So not watch it passively and observe what is happening, but actually change the behavior of cells at will. And to do so, actually, I'm interested in understanding how cells, for example, change their shape, how they acquire their final form, how also the entire organoid or tissue maintains its form and shape. And at the basis of that are cellular motor proteins that produce basically all the forces that are required to shape and to reorganize tissues. And this is what you see here. So it's highly dynamic. It's important for cell movements and cell reshaping. And we know a lot about many aspects of tissue development. But what I want to do is actually to use optogenetics to control cell conductivity and really interfere with these shaping events by activating and controlling myosin activity, so the activity of this major motor protein. And I don't want to go into any details, but basically using a two-component system that is derived from plants, I introduced this into intestinal organoids. Basically, the system allows me to enforce a protein interaction, which upon this enforced interaction, which is based on the translocation of a protein that is in the cytoplasm in the dark, translocates to the plasma membrane in the light. And at the plasma membrane, it gets active and activates myosin. And that allows me actually to do interesting set of experiments. And you see here actually a region that turns green here on the left. And as it turns green, as I shine light, actually blue light on these organoids, the organoids constrict. You see this maybe here. It constricts, and when I withdraw the light, so stop the light, the tissue expands again. And then I do it a second time, it constricts again. And when I activate a bigger region, actually this whole thing can collapse. And this allows me really, these systems allow me really to control the activity of this force-producing protein and ask questions of how that are important actually to shape organoids, entire tissues, and control, for example, the stiffness of cells, which actually paves a whole avenue of research. Another system, for example, that I'm also working on is gene expression triggered by light. So I use light as an external trigger to activate gene expression. And this is just shown here. So it's a transcription factor that gets activated by light. And having light as an external trigger actually allows us to really dynamically control signaling systems in intestinal organoids and all kind of organoids, and allows us to actually tune the level at which we want to activate the system. And we ask, for example, questions of what kind of information is actually encoded in the dynamics of a signaling system. What you see here, for example, is a signaling system or signaling pathways of the signaling pathway, and I want to ask, like, harvesting the using the ability of light to go on and off, and to deliver certain let's say laser powers, so activation strength, and see what kind of information is encoded in, for example, the periodicity of a signaling system, and also in the strength, or is it the dose that matters, or is it the time maybe at which the cells activate the signaling system. And with that, I want to finish and close my talk. And I think I want to thank Hans and this lab, and it's really a very wonderful environment. And now I think I'm already a little bit over my time, and thank you for your attention.
Well, thank you very much, Daniel, for this interesting talk. Let's see if we have some some questions. So one of the questions is, what advantages do you see? Because when you look at you're doing a light triggering, if you compare that with the classical way of activating by chemical compounds, which advantages do you see there?
Yeah, I mean, classical compounds, for example, if you induce gene expression with doxycycline or something, they actually few drawbacks. One is, or like the major one is really that it's mostly a on and off. Not like the off really basically doesn't work. So you give, for example, the compound, and as we culture our organoids in this matrix, which kind of mimics also the environment in the body, and it's actually not very. It's not. You cannot penetrate it very well. So if you once you add a chemical compound to, for example, activate gene expression, the cells will see a lot of doxycycline all of a sudden, and they will activate gene expression, and of course this compound will also will be stuck in this matrix, and even if you change the medium, it will still be there. So it's very difficult to remove these compounds once it has been added. And with light, really, we have the ability to activate at a given pulse. So we have actually the penetration depth. That's another thing that actually classical compounds lack. So we can actually go very deep. We can use 2 photons. So we have actually the possibility to go really deep inside the tissue. And of course, what I didn't talk about is actually we don't only, with light as a trigger, we can not only define very well the timing, but we can also like, we have actually all the advantages of light in terms of spatial position, right? So we can actually activate, for example, only half of an organ, half of a tissue, maybe even single cells. And this would not be possible with any of these classical techniques.
Okay. Okay. Well, thanks again, Daniel, for your talk. It's very interesting. As the last part, I would like Anna Verheer to take over and to give us an introduction into our NL5 system. So, Anna, go ahead.
Okay, thank you very much, Erdal. So in Daniel's presentation, you got a nice overview of why organoid research is important. So organoids as 3D tissue cultures gives us an excellent opportunity to examine cell behavior in its correct tissue context, which of course gives us more representative information and improved predictability. But the visualizing the organoids can be quite challenging. Organoids can come in many different sizes, from 100 micrometer to multiple millimeters, and they are all incredibly dense, which can cause difficulties if you want to image them, of course. Because it will lower the contrast and gives a high signal to noise ratio. And organoids are also very sensitive. So if you would like to do long-term imaging of the organoids, you need a very sensitive system. That's how that's why we developed NL5, which is a very fast and deep live cell imaging technology. Which allows us to use very low laser power to prevent phototoxicity. Of course, because the organoids can come in different sizes, but the hundreds of micrometer is quite thick. We need a really good technology which can give us the high spatial temporal resolution. So what is NL5? It is a confocal unit, the orange box that you can see, which couples to any inverted or upright microscopes and converts it into a highly sensitive fast scanning confocal system, which is
Anna?
Yes?
I don't see anything at the moment. I'm not sure if you're sharing.
I am checking. Can you see it now?
Yes, we just missed the beginning, I think, of the slides. But okay.
That's no problem. Yes, so as you can see, the orange box here, this is NL5, which couples to, at the moment, to an inverted microscope. And it converts it to a fast scanning confocal microscope, which is ideal for 3D live cell imaging. So this is the same image that you can see here. And as I mentioned, NL5 is a highly sensitive system. And the reason is that we use a highly sensitive camera as a detector, which allows us to use lower laser power for imaging. That's why we can do very long-term imaging with NL5. So compared to a standard confocal microscope, it has higher sensitivity and gives high resolution as well as all the confocal systems. So what for image acquisition, I am using Micromanager, which is an open source software that can control any microscope, camera, or confocal systems. But NL5 can also be controlled from any other third-party softwares. So NL5 as an upgrade that I'm controlling from Micromanager now. And it allows us to use bypass and also confocal mode as well. So if I click on the bypass mode, I already got the picture, the image of the nice organoid. This is a mouse intestinal organoid, and membrane-bound ecatharine was labeled with amnion green, which was mentioned in Daniel's presentation. So this is the bypass mode, means that I am using the epifluorescent microscope mode, which gives us a standard wide field microscope image quality. But with one click, I can switch easily to the confocal mode, which of course gives a way higher quality image. So now I am moving in Z. So NL5s around with 25 frames per second. And that's how I can do and move in into the deeper sections of the sample very quick with this confocal system. I will just come back to the middle of the image. So you can use the epifluorescent and also the confocal mode of the microscope. And also you can, after adjusting the laser power and the laser intensity, it allows you to use, of course, multiple channels. I will just quickly adjust the lookup table and improve the contrast. So this is the nucleus of the organoid. With NL5, we can also do a multidimensional acquisition. If I open the tab, which allows us to do time-lapse imaging, long-term time-lapse imaging, also multi-position. So if you have multiple wells with multiple organoids, you can just quickly scan through them with this tool. And now what we are going to do is a Z-stack imaging. For that, I will go live and then set the bottom and top position of the organoid, what I want to capture. I'm going down, so this will be my bottom position. We can also do multi-channel imaging with this system. And for time-lapse, for capturing all the important events in the cell. Yeah, sorry, I have just two lookup table. So what we can see now, we are imaging in every 15 seconds to capture all the important events we want to see. Today, I'm using a 40x oil objective for organoid imaging. Because of their density, it's really important to try to prevent the refractive index mismatch. Sorry. So if you have an upright microscope, NL5 can be coupled to it, and you can use a water dipping objective. But for long-term imaging, I recommend to use an oil objective and a low magnification oil objective for organoids, because as the organoids are growing, you want to capture a bigger field of view to be able to follow the growth and the development of the organoid. And with an oil objective, the oil is maintained longer in between the objective and the bottom of the plate. So for long-term imaging, it gives you a better resolution, a better solution. So now I'm going in Z. So I go back to my slides. So as you could see, NL5 is a really fast confocal system, which gives us high resolution, which is the same resolution as other standard confocal microscopes, so 240 nanometers in a raw image and 170 after deconvolution. It is a highly sensitive system due to the highly sensitive camera we use as a detector. And if you use the wide field mode of the microscope, then you still use the highly sensitive camera, so even you get an even improved image. Due to the high sensitivity, we can prevent phototoxicity and bleaching of the fluorophores. About the application, so due to its fast scanning speed, it allows us to study fast live cell dynamics. This is a nice HeLa cell, which is currently sensing the environment. You can see the GFP. So you can study cell signaling in intra and extracellular transport. It is ideal also for long-term live cell imaging. This is a zebrafish embryo, which was imaged with a 10x objective to capture all the information we need. And it was imaged for more than 24 hours. You can see the nice endothelial cells. And even after 24 hours, there is no sign of phototoxicity. For this experiment, we was imaging a Volvox sample, and now we are going deeper and deeper in the sample. And even if we went, we are now more than 90 micron deep in the sample, we got a nice high contrast image with this confocal unit. Due to its scanning speed, it is ideal for high content screening as well. So for example, for drug discoveries. And the color switching is not a problem. So you can just quickly scan through the whole 96 well plates with multiple positions, multi-channeled, and you can image them in Z. Thank you very much.
Well, thank you very much, Anna. To give us your introduction on NL5. I'm just checking if we have some more questions. I don't see any further questions. And we anyhow also running a little bit out of time. We're almost on the 45 minutes. So in the meanwhile, if you do have questions or you would like to ask them, please contact us directly via our website or via sales@confocal.nl. And hope to see you back at our next webinar. Thank you very much for joining.
Thank you. Bye.
Bye.
Bye-bye.